One of the things which is really neat about this research program is that it's not just the output at the end, which is of scientific interest, it's how we got there. That layering of information from different disciplines, from the first leg and the second leg, and now our biodiversity focus in the third leg, to say something meaningful about the reefs. My name is David Aguirre. I'm one of the primary investigators on the Inkfish Coastal Seas Mesophotic research program, and we're currently here on the final leg of our expedition in Tonga. The research that we're doing is within the Ha'apai region, where relatively little is known. As a whole, this expedition is focused on mesophotic reefs. Those are the reefs that occur from 150 metres right up into the light-filled shallows. The third leg of this expedition is primarily focused on different organisms that occur across that large depth gradient. Up in those really shallow parts of the reef, we go and do the biological collections of certain harvested species to tell us a little bit about how they're connected to different islands around the South Pacific. Clams are of particular interest because they are an indicator for climate change. We take small tissue samples from them to do the genetic analyses. Once we find out what happens to them, we can extrapolate those findings out to similar species in similar parts of the world and tell a little bit about what might be happening at a global scale from results found at this local scale here in Tonga.
Okay, G1 is recording.
For the lower parts of the mesophotic reef, we've got the beta remote underwater video surveys.
Yep, good.
Which is a completely non-invasive method, telling us about the fish communities here in Tonga.
Bottoms. Bottom.
The goal for the BRUV data set is to identify and measure every fish that we've seen, which is really important for fisheries management. Yes, jobfish. What we can then do is link any changes that we're seeing in abundance, fish biomass, even fish behaviour, with the data that's been captured before us. So changes in the water column, on the habitat, on the seafloor, and get a complete picture of what's happening in the mesophotic range.